Good afternoon, everybody. Welcome to the next seminar in the INTERMICRO series. And it's my pleasure to welcome Zuzana Kalaninová, who will speak about some enzymes in fungi. Mm-hmm. The floor is yours.
Yeah. Thank you for the kind introduction. Hello, ladies and gentlemen. Today I'm going to talk about one fungal protease, which is known for quite some time, but we still get to discover something new about it. Every biological process is governed by proteins, and to understand their composition in cells and in organisms, and to understand their modification functions, structure is crucial to understand their role. And out of many techniques available, mass spectrometry has become one of the most powerful tools, providing unique information about dynamics, about its sensitive technique, and we can study also the highly complex samples. Proteomics distinguishes three main approaches: top-down, middle-down, and bottom-up. In bottom-up approach, we took protein, and we take protein and we digest it into small peptides, which are then analyzed by mass spectrometer, and the data are searched against a database. Solution digestion is performed either in solution or online using enzymatic reactors with immobilized protease. For in-solution digestion, typical first choice is trypsin, which cleaves specifically after at the C-terminal of lysine and arginine. Despite its numerous advantages, it struggles with some cases where natural distribution of lysine and arginine is unfavorable, and resulting peptides are either too small or too large for identification, or if there is proline at stop site. At P1 position, which is stop site. Then this all fueled quite extensive research on novel proteases. And one class of these alternative proteases are proline-specific enzymes. Their story began in 2009 with identification of proline-specific protease from Aspergillus niger fungi, and proposing its use in proteomics. In following years, another proline-specific enzymes from pitcher plants were studied, and in 2017, on the Aspergillus niger prolyl endoprotease, let's call it ANPEP, it was successfully used in hydrogen deuterium exchange mass spectrometry to study protein structure. Here we got inspired, and we prepared ANPEP-based columns and used it to study bank wall prion protein. This protein is challenging analytical target because of its unstructured N-terminal rich in repetitive sequences and in proline. As you can see, we failed with pepsin, typical first choice for online digestion, and this problem was solved with employing the ANPEP-based columns, either with ANPEP alone or in combination with, for example, pepsin or nepentazine 2. Then, a few years later, research-grade ANPEP called proalanase was used as an effective alternative to trypsin for different proteomics applications, and conditions for specific digestion were described. These were 1:50 protease to protein ratio at 2 hours reaction time and pH 1.5. So we wondered how specific is our column. We prepared simple protein mixture consisting of 5 proteins differing in size and proline content. And we tested also the effect of temperature and flow rate. But as you can see, our column was far from being specific. The question remained why. There are several factors that we try to consider and think of. And the first one was complexity of sample. We used UNPEP column to digest a human serum, and as you can see, complexity really matters because cleavage profiles were different if I know. That's amazing.
No.
No? Everything good?
Yeah. I think he.
Okay. Okay. So, and you can see the clear difference in digestion profiles based on all proteins identified in human serum. When we looked into cleavage preferences of only the most abundant protein, human serum albumin, and when we removed this most abundant protein. Then we more? No. Perfect. So another thing we wonder if could be behind the unspecificities was whether there is any contamination in our source of protease. So we performed proteomic profiling of UNPEP, and as you can see, UNPEP was the main component. It's the one in light blue shade. And this was also in agreement with SDS page profile. So now we know that complexity matters and that we do not have any contaminants present. So another thing, or another two factors we could think of was the source of our protease and the concentration. Because when we are digesting online, the whole proteolysis step takes only 3 minutes. So in order to be able to quantitatively digest our sample, there needs to be high concentration of our protease. So we took the same protein mixture and we tested two sources of protease, industrial grade UNPEP and research grade prolanase. Here you can see the difference in price and the reason why we prepared our column only with the industrial one. We studied the effect of time and the effect of protease to protein ratio, and we saw the trend that with increasing time and with increasing ratio, the specificity decreased. Basically, the longer the time and the more the protease you have, the less specific is your digestion. So now we know that the reason why digestion of on our proteolytic column with UNPEP was not specific. But another thing that caught our eye here was rather high specificity for cysteine, which was never described before, nowhere. We then performed an easy experiment where we took our protein mixture and we reduced it, or we reduced and alkylated it. And as you can see, after alkylation, so after modification of cysteine, there was a huge decrease in activity. So we tested this also on more complex sample on cell lysate. We further tested this post-cysteine cleavage theory on a simple analytical target on human insulin. It has 2 chains and only 1 alanine, 1 proline, and multiple cysteines. We tested it on non-reduced, reduced, and on a beta chain with oxidized cysteines, and we got to the same conclusion that ANPEP is able to cleave after cysteine only if it's in reduced form, and whether it was incorporated in disulfide bond or it was somehow modified here with oxidation, there was no cleavage after this residue, which proposes another great use of this enzyme as a tool to validate cysteine or disulfide bond and modification status. Next, we wondered if it applies also to other postproline cleaving enzymes. We took neprosine. It's a protease found in fluid of pitcher plants, and it belongs to completely different protease family. Its cleavage profile is similar to the ANPEP, and we got again the same results. So neprosine is able to cleave after cysteine only if it's in its reduced form. If there is any modification, there is no or minimal cleavage. We put all this our knowledge and more in this publication last year. So to give you some take-home message, the postproline cleaving enzymes cleave after cysteine, which means that we should probably reconsider many things we know about proline-specific enzymes, like their mechanism, therapeutic role, or possible new applications. Modifications like alkylation, oxidation, or incorporation in disulfide bonds inhibit the post-cysteine activity, which makes these proteases good tool for validation of disulfide bond status or modification status, and propose its use, for example, for quality control of antibodies. And ANPEP is very sensitive to digestion conditions, and because of that, it probably will never be specific on a column, but it doesn't mean that there is no use for these columns, because as you could see in, for example, for the prion protein, we would be lost without it. And with that, I'd like to thank all my colleagues and funding agencies and you for your attention.